Hello student, welcome back to the next topic of the cell, the unit of life. So, in this video we will study ribosomes and inclusion bodies. Ribosomes. In prokaryote, ribosomes are associated with the plasma membrane of the cell. They are about 15 nanometer by 20 nanometer in size and are made up of two subunits, that is 50S and 30S. So they have two subunits. One is large subunit and one is small subunit. So units which when present together form 70S prokaryotic ribosome in prokaryote. Ribosomes are the site of protein synthesis. So here on the ribosome, the protein synthesis takes place. Several ribosomes may attach to a single mRNA and form a chain called polyribosomes or polysomes. Okay? So ribosomes get attached to the mRNA strand and forms a chain called as the polyribosome or polysome. The ribosomes of a polysome translate the mRNA into protein. So this mRNA is translated into proteins by ribosome. Now let us see the difference between prokaryotic and eukaryotic ribosomes. So ribosomes are present in both prokaryotic and eukaryotic. Now prokaryotic ribosome has 70S ribosome with 30S small unit and 50S large unit, whereas in eukaryotic ribosome it is 80S ribosome, whereas 40S is small unit and 60S is a large unit. Now S is the sedimentation coefficient. Now here in 16S there will be 16S rRNA plus 21 proteins, whereas in 50S there will be 23S plus 5S rRNA plus 34 proteins. So in eukaryote 40S small unit you will have 18S rRNA plus 30 proteins, and in 60S 28S 5.8S plus 5S rRNA plus 40 proteins. Now this is the 30S, 50S, these are the numbers wherein the mRNA gets sedimented, okay, mRNA gets separated. So that is the sedimentation coefficient. So you cannot add 30 plus 50 and you won't get 70S. It is a sedimentation coefficient. Similarly here 40 and 60 you won't get 80S. It is not addition, it is a just sedimentation coefficient wherein they get separated. Now inclusion bodies. Reserve material in prokaryotic cell are stored in the cytoplasm and in the form of inclusion bodies. Okay, so the reserve material, whatever the store they want to store in the cytoplasm, is stored in the form of inclusion bodies. So you can see here these are the small granules like structure which are present in the prokaryotes. Now these are not bounded by any membrane system and lie free in the cytoplasm. They do not have membrane bound system. They are free in the cytoplasm. Examples of inclusion bodies are phosphate granules, cyanophysin granules, and glycogen granules. Gas vacuoles are found in blue-green algae and purple and green photosynthetic bacteria. Okay. So these are blue-green algae and these are purple photosynthetic bacteria. Now eukaryotic cell, let us see the structure of eukaryotic cell. The eukaryotes include all protists, fungi, plants and animals. Okay, so the eukaryotic begins with the protists. So they are the first one to get the nucleus. So protists, then fungi, plants and animals. These all are eukaryotic organism. Whereas protist is the unicellular eukaryotic organism. Now cytoplasm contain membrane bound organelles. So you can see all the organelles are they are bounded by the membrane, another membrane too. So inner membrane and the outer membrane. So that's why they are called as the membrane bound organelles. It possesses an organized nucleus with a nuclear envelope. So you can see this is the nucleus with nuclear envelope which is absent in the prokaryotes. It has a variety of complex locomotory and cytoskeletal structure. Their genetic material is organized into chromosome. So you'll find inside the nucleus chromosome wherein genetic material is present. All eukaryotic cells are not identical. Plant and animal cells are different. So although these are in eukaryotic cell, but they are not identical. You will find difference in the plant cell and the animal cell. So plant cell and animal cell find it. So this is the structure of the plant cell. Now plant cell consists of nucleus, a big vacuole, then lysosome, rough endoplasmic reticulum, chloroplast, mitochondria, ribosomes, cytoplasm, smooth endoplasmic reticulum, then Golgi apparatus, nuclear envelope, plasma membrane. This is the adjacent cell wall. So this is the 3D structure of the plant cell. Now animal cell. Sorry. Animal cell consists of Golgi apparatus, microvilli, plasma membrane, centriole which is absent in the plant cell, then cell wall is absent here, peroxisome, lysosome, ribosomes, mitochondria, rough endoplasmic reticulum, then cytoplasm, nucleus, nucleolus, nuclear envelope, smooth endoplasmic reticulum, etc. Now let us see the difference between plant cell and animal cell. Plant cell has a distinct cell wall as a outer boundary and within it the cell membrane. Whereas the animal cell has an outer membrane and cell wall is absent. The plant cell lacks centriole. Animal cells contain non-membrane bound organelle called centriole which helps in cell division. Plant cell possess plastids and a large central vacuole. Plastids and a large central vacuole are absent in animal cell. So these are the difference between plant cell and animal cell. Now cell membrane, the next topic is the cell membrane. Cell membrane. Cell membrane is composed of lipids that arrange in a bilayer. I'll show you in the diagram. Lipids are arranged within the membrane with the polar head towards the outer side and the hydrophobic tails towards the inner part. So this is the lipid bilayer. So this is present on the cell membrane. So these are the polar heads and these are the tails. So polar heads on the outer side, outside of cell membrane, and this lower side is inside of the cell membrane. So this remains outside the cell membrane and this remains inside the cell membrane. Okay. So lipids are arranged within the membrane with polar head towards the outer side and the hydrophobic tails towards the inner part. The non-polar tails of the saturated hydrocarbon is protected from the aqueous environment. The lipid component of the membrane mainly consists of phosphoglycerides, proteins and carbohydrate. The ratio of protein and lipid varies in different cell types. In human beings, the membrane of the erythrocyte, that is the blood cell, has approximately 52% proteins and 40% lipids. That means the lipid and protein ratio varies from different cell, that is from human beings and other animals. It depends upon the which cell you are taking, whether it is plant cell, animal cell or human being. So membrane proteins can be classified as integral or peripheral. Now you can see integral protein are those proteins which are found in between the lipid bilayer, whereas peripheral proteins are present on the outside of the polar head. So those are called as the peripheral proteins. Peripheral proteins lie on the surface of the membrane, while the integral proteins are partially or totally buried in the membrane. Cell membrane was proposed by Singer and Nicholson in 1972 and also called as fluid mosaic model. Okay. Now you can see this is the structure of cell membrane or the fluid mosaic model or the plasma membrane. So you can call it by three names: cell membrane, plasma membrane or fluid mosaic model. Now this is the phospholipid structure. So you will have hydrophilic head and hydrophobic tail. That means it repels water. Okay, hydrophobic tails repel water. So it is found in the aqueous environment. Now these are the polar head and this is the non-polar region. So this is the polar region, both the ends, and this is the non-polar region. Now these are the integral proteins which are embedded in the protein bilayer. Okay, now these are the cholesterol unit. This is the protein channel which helps in the transport of protein. Also called as the transport of molecule. Now these are the peripheral protein which lie on the surface of the lipid bilayer. Now these are the hydrophobic alpha helix protein. Now this is the globular protein. These are glycoproteins. And these are the carbohydrate chains. And these are the glycolipid chain. Okay. So this is the structure of the cell membrane. So you have, there are two different types of protein are present: integral protein and peripheral proteins. Then glycolipids, carbohydrate chain, glycoproteins, cholesterol, etc. Lateral movement of proteins within the bilayer is measured as its fluidity. Now protein molecule can move, or the lipid bilayer can move laterally. This is called as the lateral movement, which is frequent. You can see here within the lipid bilayer, and this is the flip-flop movement of the lipid molecule, which is very rare, whereas this lateral movement is frequent, and that's why it gives fluidity to the bilayer. Now functions of the lipid bilayer or the membrane. The fluid nature of the membrane perform functions like cell growth, formation of intercellular junction, secretion, endocytosis and cell division. Plasma membrane transport molecule across it. The membrane is selectively permeable to some molecule present on either side of it. So that means it will allows only certain molecules to pass through it. And the protein molecules helps acts as a channel in passing the molecule. The molecules which can move across the membrane without any requirement of energy is called as the passive transport. So molecule when directly move inside the membrane across the membrane, it is called as the passive transport. Now neutral solutes move across membrane by the process of simple diffusion along the concentration gradient, that is from higher concentration to the lower concentration. Water also moves across this membrane from higher concentration to lower concentration, and such movement of water by diffusion is called as the osmosis. So what is osmosis? The movement of water molecule from higher concentration to lower concentration by diffusion process is called as osmosis. So osmosis is the term used for water movement by diffusion process. Now, polar molecules cannot pass through the non-polar lipid bilayer. They require a carrier protein of the membrane to facilitate their transport across the membrane. A few ions or molecules are transported across the membrane against their concentration gradient, that is from lower to the higher concentration. Such a transport is an energy dependent process in which ATP is utilized and is called as the active transport. Example, sodium potassium pump, wherein ATP is required to transport the molecule. Now let us understand in more by watching this video.
Are enclosed in a plasma membrane, which consists of two layers of phospholipids. The nature of the cell membrane makes it intrinsically permeable to small non-polar and uncharged.
So I hope you have understood by watching this video So in this video, they have explained how the passive and active transport take place. And also you have seen how the protein acts as a channel for transporting molecules. Okay, so it is very clear how the membrane works by watching this video. I hope you have understood today's topic. If you have any difficulty, let me know. You can comment. Thank you, students.